Okay, hello everyone, and welcome to the next video in the DNA to protein series for AS level biology, or full A level, depending on where you are. We're going to get into the details now about exactly what goes on in these processes between the DNA and the polypeptide. Okay, so we've already done an overview of this, a brief overview of the idea with the genetic code, and the idea that DNA is sort of copied out into RNA, and RNA then is read and translated into protein. Okay, but we're going to go into the details of the transcription side and this process of splicing and modification. And this lesson is going to come in three levels. If you've not seen one of my biotech or high-level biochem lessons before, they come in various levels. We're going to increase the difficulty and the complexity as we go through, from a basic answer of how this process works up to something a bit more nuanced and a bit more detailed towards the end. Okay, so this is the bit of syllabus we're looking at here. The idea of transcription being the production of mRNA from DNA, the role of the different enzymes involved in that, and how it's different in prokaryotes and eukaryotes. Now, as we get bogged down in the detail, as we're going to get really kind of into the detail here, I want you to keep an overview in your head of what we're actually talking about. And that's this picture here, which I've shown you before, right? Which is basically the overview. So all the information is here on our red DNA. That's the master copy. This blue stuff is the RNA for the molecule that wants to be moved out to the cytoplasm to be read and made into protein. And then the ribosome here, of course, is the organelle that's actually going to do the work of stitching these amino acids together and with the help of tRNAs and manufacture that finished protein. Okay, so in this video we're going to go through the stage that's here in the nucleus. So basically everything from the DNA up to where this black arrow shows the RNA shooting out through the membrane. Okay, so as a little recap, again I've shown you this flow diagram before in previous videos. The code for the proteins on the is in the DNA bases. That then gets transcribed to this stuff called pre-mRNA. Pre-mRNA is spliced to form mRNA, and then that is what gets translated. So it's this first bit here, these first three arrows, that we're going to talk about now. So by the end of the lesson, by the end of this video, you should know what all of these numbers mean. So basically with that picture there, and again, you can sort of find a picture like this online or take a screenshot or whatever. We're going to come back to this picture again and again. You should know what's happening at 1, 2, 3, 4, 5, 6, 7, and 8. Okay, so you should know what all of those stages are. And if you don't recognize what's going on at one of those stages, then go back, look at that bit of the video again. If you still don't understand, get in touch. Okay, and I'll try and clarify it. So we'll start then, right, with the simple version. Now this is our basic version of how transcription works. Okay, at its sort of simplest level. Okay, what it produces is stuff called pre-mRNA. It's also referred to as the primary transcript, which is then modified to make the mature mRNA. Okay, this all happens in the nucleus, of course, because that's where the DNA is. Okay, now here's an enzyme that you've come across before. Okay, DNA helicase breaks down hydrogen bonds, so it basically splits the DNA helix and creates this gap here, right, which we call the transcription bubble. Okay, so that's the same enzyme that's in DNA replication. Okay, you will have come across that before then. Now we've got a different but similar to one you've come across, right? Because this time we're not trying to make more DNA, we're trying to make more RNA. So if you remember your DNA replication, you'll remember the other enzyme was called DNA polymerase. So it will surprise nobody to realise this one's called RNA polymerase. But I really have to stress, because this is a mistake that gets made year in, year out in test and exam answers, what polymerases do is they manufacture polymers. Okay, they don't stick the hydrogen bonds together. Okay, that is not what they do. They don't stick the hydrogen bonds together. The hydrogen bonds form passively. The bases diffuse in, and by complementary base pairing, they stick to their complementary base. Okay. The RNA polymerase's job is to come in and join the backbones up, or join the phosphodiester bonds up between the sugars and the phosphates, not the hydrogen bonds. Okay, very important to get that clear. So this is called a transcription bubble, what you find is there's 12 base pairs unzipped roughly, approximately at a time, called the transcription bubble. Okay, and this starts at what we call a promoter region. Right, that's how RNA polymerase knows where to bind on. There's a special region in the DNA which is like a signpost saying gene here, transcribe this. And if you want more information on how that works, that's at the A-level standard, so you can look at the more advanced gene expression playlist, which will explain all about the regulation and control of those. Okay, so promoter and terminator regions tell RNA polymerase where to start and stop. Okay, so if we go over to the diagram and go through this step by step, first thing that happens, DNA is unwound by DNA helicase. Now in the nucleus, we've got a supply here of RNA nucleotides, okay, which are going to diffuse in passively and form their hydrogen bonds. I repeat, passively, no enzyme required, okay, not what RNA polymerase does, okay. So what we're seeing here, look, as this bubble moves along, okay, our RNA is being synthesized following on one strand. Right? As we've said before, we call that the coding strand or the sense strand. Okay, that is going to have the information for making protein on. This other side we call the non-coding or antisense strand, and that is just there for replication, as per the previous videos on replication. So this red strand here is the one that's being used. Okay, the one that's being used. RNA polymerase is going to follow up that strand here, right? So complementary RNA nucleotides are attracted and base paired to the coding strand of the DNA, and then RNA polymerase comes along and joins them all up. Okay, number 3, look there we've got, so our 3 nucleotides are joined together by the RNA polymerase, phosphodiester bonds form between them. Remember that is the backbone and not to the other strand. Okay, if we had phosphodiester bonds to the other strand, you'd never get them to come apart. Right, and then lastly, as this moves along, right, not many of them are actually stuck on at any given time, 8 to 10-ish, and they peel off as the strand rewinds. So as the strands rewind, this primary transcript, this pre-mRNA, sort of peels off behind. And that's the basic process of transcription. Okay, starts at a promoter. You get a transcription bubble made by helicase moving along, the RNA nucleotides diffuse in, and polymerase comes along and sticks them all together. Right? The DNA winds up behind the RNA polymerase and pushes out the primary transcript, and that's your pre-mRNA. Okay? So that's on our diagram. I mentioned that's our first sort of 3 steps of what happens. Okay? Now, in a prokaryote, in a bacteria, which these pictures are 2 different colour versions of the same picture, but this is from a prokaryote, that's pretty much it. Right? Now, this is an amazing bit of immune fluorescence microscopy going on here. So we've used some fluorescent dyes and some antibodies to stick to different things, to stain them different colours. Down here, look, the pink here, or the blue here, that's the DNA. All this transcription is going on here, right, in these little bits, because the yellow in this picture, or the red in that picture there, that is the RNA being produced, and then all of these blobs, blue over here, green over here, are ribosomes. So literally the ribosomes which do the next bit, translation, have just piled on and just started translating immediately as the DNA is being transcribed. Okay? So in prokaryotes, this version here, right, is all that there is, right? There's no difference between pre-mRNA and mRNA. The primary transcript just gets translated and that's made into protein, job done. Okay? So that obviously is because they don't have a nucleus, so there's no difference in place, in location, or whatever it might be. Okay? Now, in a eukaryote, it's a bit more complicated. That pre-mRNA has some processing to do. So we're going to lift this up another little level, right? So that first part, that's our sort of basic level. Right, and now we're going to lift this up to another level where we explain specifically how eukaryotes go and add this idea of splicing in. So within a gene, right, within the primary transcript, there'll be bits that are destined to code for amino acids, and there'll be bits that aren't. Okay? The bits that code for proteins we call exons, and the bits that get cut out and aren't used we call introns. Okay? And those are words on your syllabus, you are expected to know them. The process of doing that cutting is called splicing. And what happens, if you're keeping an eye on that first picture, is that the two ends of the exon are kind of pulled together and the intron's looped up and it's then cut out. Okay? But it's done by a combination of proteins called a spliceosome, because biologists are not very good at naming stuff. Okay? Then after that's happened, that's when you've got your mature mRNA, and that leaves through the nuclear pore. Okay? Or at least that's the case until we take it up to the third level of difficulty. Right? So here we've got our primary transcript. So this is straight off the DNA. Okay, straight off the DNA there. In this instance, look, we've got 2 exons and 1 intron. We want to stick these exons together. Okay, they're the bits we're interested in. So our spliceosome, right, which is made of a bunch of different proteins, usually that's what osome means, a complex of proteins, comes together, pulls the end of these together, makes like a P shape initially called a lariad, right, and then you get cutting at the individual pieces. Now I want to recommend another YouTube channel here, which is called the Vcell collection, Virtual Cell Collection, which are really, really good at animating all these kind of processes. Okay, so if you want to see this animated really nicely, then I would recommend the Vcell collection. I'll put the link in the description. Okay, so once we've got that, right, there's our little lariad, our little P-shape bit of RNA that's cut out, and these 2 exons are stitched together, and they're our mature RNA. Okay, so in prokaryotes, this doesn't happen, goes straight from the basic transcription straight on to translation. In you, there's this mRNA processing, this splicing in eukaryotes, where we cut out the useless bits. Now you might be asking the question of, well, why are they there? Well, as we discussed last time, we talked about non-coding DNA, there's loads of possible reasons. Most likely here seems to be viral insertions over history. The idea that throughout our evolutionary history, our DNA has been bashed about, it's been altered, it's been changed, it's had viruses attacking it, it's had bits that have had mutations go on, and you never get an overview, you never get a designer coming along and going, hang on a minute, we don't want that, we'll get that, we'll just tidy all this up here. You just end up sort of making, doing, carrying on. That's how the evolutionary process works. Okay? So that is kind of the explanation here, and as far as a better explanation that goes, I think it would vary gene by gene, right? And obviously you can go and do a bit further research there if you want to know a bit more about the origins
So, are you
of
there?
the exons. Okay? So here we have our polymerase. Right, here we have our polymerase at number 1 and number 2. Right, this is number 3 here, number 4. Okay, we've got number 5 is our splicing bits, that's our pre-mRNA. Number 6, our introns are spliced out. Number 7, that's our mature mRNA. Number 8 here, we've got our export then of our mature mRNA through the nuclear pore. Okay, so that's basically where you need to be. We're going to take this one more level up in terms of one more level of difficulty. Okay, that's to say there are some other modifications that occur to the mRNA before it goes out. And these are super clever, right? This is why molecular biology is so interesting, right? These are super clever ideas. So essentially another level up, this bit here, the red bit, right, and the pink bit, we've already discussed that. So there's a load of exons there, we've got 2 introns, they get spliced out. But there's 2 other features on this picture here, look, and that's the M7G cap and the poly A tail, which get added on post-transcription. Okay, so first thing that happens is a methylated cap gets added to the 5' end, and that looks like that. Okay, so this is standard RNA, as you can see, lovely chain there, nice and ordered. And then this mangled, garbled thing is basically, look, a backwards base. Okay, and it's a heavily methylated version of the G base gets added on backwards. Now the reason for that is when RNA enters the cytoplasm, there are a load of enzymes that are immediately going to want to break it down. And that's good, because you switch a gene on, you want also something to be able to switch it off. And that's nuclease enzymes. Okay? Now you want to have some level of control. So this methylation, this adding a backwards base with all sorts of methyl groups on the end, makes it nigh on impossible, right, for an enzyme, a nuclease, to get onto the 5' end and start to break down the RNA, right? So the 5' end is pretty well protected from these nucleases. which that's good because our ribosome can then start attaching and manufacturing copies of the protein. On the other end, okay, you've got this thing called a 3' poly A tail, which is basically just where a load of A bases are added, a load of A nucleotides. So basically you just loads of A's. And they can be attacked by nuclease enzymes. Okay, so the nuclease enzymes will come onto the 3' end and start chomping away. And once they chomp their way through the poly A tail into the coding bit, well then translation of that protein will stop. That's the off switch. Okay. Now, if this is a protein you want expressed for a long time, right, then you'll probably want to have a long poly A tail on. So it takes a long time for the nucleases to chomp through it and turn that gene off, turn that mRNA off. If you want this only on for a short time and then off, like a really quick responsive protein, you'll put a very short tail on. So chomp into the coding region, protein is stopped being produced. Okay, so it's super clever. I sets the timer like a fuse. Because this depends. This is a way of modulating and regulating how DNA, how long a particular piece of DNA is expressed for, and it's a super clever method. So the methyl cap stops attack from the 5' end. The poly A tail stops chomping away from the 3' end. Okay, so we call these processes post-transcriptional modification: splicing, methylated capping, and polyadenylation on the tail. Okay, so that now should then cover all of those numbers. 1, 2, 3, 4, 5, 6, 7, 8. You should know at a level of detail that suits you what is going on at each of those numbers. If you don't, then run the video back to where that is and have a look. Okay? So in terms of comparison, this is a question you can get. Compare transcription DNA replication. We've got our green bits there as similarities and our red bits there are differences. Okay? So I would suggest taking a moment to read that. Of course, the fact helicase exists. They both occur in the nucleus. They coil up afterwards. They recoil. So that kind of stuff to make a double helix afterwards. But then obviously you've got some major differences, particularly in the polymerases and in the product in terms of what you get and then how many strands are used. Okay, could be a question that you could get. Do a comparison. So it's worth pausing the video here, have a good read, make some notes if you see fit. Okay. Now I'm going to do something I've not done before because this is for part 1 of a 2-part lesson. I'm going to give you a query at the end. I'm going to give you a question. Okay, so let's imagine a mutation occurs that destroys the stop codon. Okay, what amino acid would the resulting mutated protein be rich in and why? Okay, so you will read that again. You've got a mutation that destroys the stop codon here. Okay, now if you don't remember what a stop codon is, look back to the genetic code lesson. That's in that one. Okay, so your ribosome running along here, reading this, that is now destroyed. Which amino acid, what amino acid would the resulting mutated protein be rich in and why? Answer will be in the next lesson. Thanks very much. See you then.